So now we're going to be talking about the second cell division, also known as meiosis. Only very specialized cells undergo meiosis, that is, the egg and sperm are the only two cells that undergo meiosis. Now meiosis and mitosis are similar in certain ways and also different. So in here, I want to focus on the differences between meiosis and mitosis. Alright, so first we must learn some vocabulary. So alleles are different versions of the same gene that have different nucleotide sequences. Diploid organisms have 2 copies of every single chromosome. Homologous chromosomes are a pair of chromosomes that contain the same genes. They contain the same genes, but they might have different alleles. And if you guys remember, alleles are different versions of the same gene. In a diploid cell, one homologous chromosome is inherited from the mother, and the other one is from the father. So we are a perfect combination between our mom and our dad. We are not identical. We are a perfect combination between mom and dad. Genotype is the genetic or allele makeup of an individual, whereas phenotype is the observable or measurable features of an individual. So the genotype determines your phenotype. So phenotype, think about what you can actually see, your looks, if you will. So genes provide the instructions for making proteins. So genes are physically transmitted from parent to offspring. So in a diploid organism, we have 2 copies of each chromosome. These are known as homologous chromosomes. One comes from mom, and the second one comes from dad. So sexual reproduction is the combination of maternal and paternal alleles. So those 2 cells are joined during fertilization, and it determines the genotype, and it also contributes to the phenotype. Sexual reproduction requires specialized cells known as the egg and sperm. Another term for the egg and sperm are gametes. So gametes are basically cells that have half the number of chromosomes, and we produce those gametes through the process of meiosis. So meiosis is a special cell division that produces genetically unique haploid cell. So whenever we want to make an offspring, so what happens is the egg and the sperm, with both of them are haploids, which means they only have 1 copy of each chromosome, they come together to make a complete organism, an organism that just has the right number of chromosomes. And so the result is what we call a zygote. And then the zygote is going to divide by the process of mitosis until we actually have an embryo, which is basically an organism. So meiosis consists of 2 separate division steps. So just like mitosis, you're still going to have the reproductive phase or interphase where you have G1, the S phase, and the G2 phase. And then you're going to have, prophase, metaphase, anaphase, telophase 1 of meiosis 1. And then you're going to have prophase, metaphase, anaphase, telophase of meiosis 2. So overall, what happens during meiosis 1 is that the homologous chromosomes are separated. Each dado cell is a haploid cell, and each chromosome still has 2 sister chromatids. So each of those sister chromatids are still attached to the centromere. So during meiosis 2, what's going to happen is we're going to separate those cells into individual chromosomes. So overall, what happens in meiosis 2 is that we separate those sister chromatids. So that way we actually have 4 cells that have half the number of chromosomes than the original cell. And those are the ones that are going to develop into the egg or sperm. So I want to point out 2 very important processes that happens during meiosis. So during prophase 1 or meiosis 1, what happens is that the chromosomes are basically touching each other. And everywhere they're touching, they're actually exchanging genes. So they're recombining, if you will. And then the second thing that happens is the alignment of the chromosomes. So the alignment of the chromosomes in the middle during metaphase is going to determine what cell is going to end up with what chromosomes. And that's important for increasing diversity. So one of the things about meiosis is that at the end, the cells are going to look different than the parent cells. They're going to look different in terms of the amount of chromosomes that it has. So it's going to have half the number of chromosomes than the parent cell. And it's also going to look different in terms of the combination of alleles that are found within each chromosome. So overall, I basically say that meiosis is important for diversity. So overall, meiosis is important for genetic diversity. So no two gametes are identical. And the reason why no two gametes are identical is because of the process of recombination or crossing over and independent assortment. So overall, meiosis is important for genetic diversity. So recombination is whenever the maternal and paternal chromosomes are physically touching each other and they begin to exchange alleles. So this happens during prophase 1 or meiosis 1. Independent assortment is alleles of different genes are distributed independently of one another. And both of those, recombination and independent assortment, increases genetic diversity. And this happens during meiosis. So the purpose of meiosis is basically to create cells that look different than the parent cell. They look different in terms of the amount of chromosomes that it has and also the combination of alleles.